Welcome to Crunch Academy. So, hope everyone is doing good. Today we'll be continuing on our neurophysiology series, and this will be part 3. Those who haven't watched the part 1 and part 2, I request you to watch those so that you can get a better understanding of what I'm speaking today. By the way, Eid Mubarak to all my Muslim friends out there. So, in my last 2 sessions, I've been keeping on saying that information is always carried from the peripheral nervous system to the central nervous system, and from the central nervous system to the effectors. Right. So, who carries this information? In what form are these information carried? Many of you might have answered this now. The informations are carried by the nerve cells, and the information is carried in the form of electrical signals, right? So, is it whether a single nerve cell or the single neuron, or there are multiple neurons? If there are multiple neurons, how is information transferred from one neuron to the another? Okay, so we'll be answering these questions in today's session, and I would like you to know one thing. Those electrical signals or the information that is carried by the nerve cells are referred to as action potentials. Before we move on to action potentials, let me give you a brief idea on what are membrane potentials. Okay, so it will be easier for you to understand what are action potentials, or you would easily understand the concept of action potentials. Here, I will be drawing two cells, two hypothetical cells. Okay, this is 1, this is 2. As you know, every cell has its own nucleus. So we have a nucleus here. We have the DNA within it. And this DNA is the reason, or this DNA encodes for different proteins present on the cell and outside the cell, right? So one such protein that the DNA of a cell encodes is this one. Okay. And the function of this protein is to act like a pump. And what does a pump usually do? It takes in something and it pushes out something, right? Consider the cell to be in an ocean. Okay, and the ocean is always rich in salt. Salt chemically is sodium chloride. So every cell, or outside every cell, we have sodium and chloride in abundance. So we have sodium in abundance outside the cell. And what is the element that is abundance within the cell? That is definitely potassium. Okay. So we have an abundance of sodium outside the cell and abundance of potassium inside the cell. Okay. Now, what does this pump do? This pump acts against the concentration gradient. Means sodium is in abundance outside the cell. It is less inside the cell. Similarly, potassium is in abundance inside the cell, whereas the concentration of potassium is lower outside the cell. This pump acts against that concentration gradient. It tries to push, or it tries to take sodium out of the cell and bring potassium in the cell. Okay, so it is trying to push more and more sodium into an area where there is more sodium in abundance, and it is trying to take in potassium where already the concentration of potassium is too high. Okay, since this is acting against the concentration gradient, it definitely requires some energy, and this energy is provided by the ATP present in this pump. It phosphorylates this ATP into ADP and phosphate, and it uses this energy to pump out sodium and pump in potassium. Now, this pump is a little biased. It does not push out the same amount of sodium as it takes in potassium. It always pumps out 3 sodium for every 2 potassium taken in. Okay, for you to remember it easily, just remember this decade. It is always 2000, right? Or in Gen Z way, we say it 2K, right? So 2K means 2 potassium. 2 potassium is always taken inside for every 3 sodium pushed outside. Okay. The action of this pump is to take in potassium and push out sodium with the help of ATP. And thus this pump is referred to as the sodium potassium ATPase. Okay, sodium potassium ATPase. Now, just think, it pumps out 3 sodium for every 2 potassium taken in. Okay, so there is a deficiency of one cation. Cation means positively charged particle. Both sodium and both sodium and potassium are positively charged particles, right? So for every 3 sodium taken out, it takes in 2 potassium in. Means there is a deficiency of one cation which is present inside the cell, right? So the cell starts to have a slight electronegativity, right? Electronegativity due to the action of the sodium potassium ATPase. Now, the same DNA which encoded for the sodium potassium ATPase is also responsible for another channel which is referred to as the leaky potassium channels. And as the name suggests, leaky potassium channels, they help in the transfer of potassium from a region of higher concentration to a region of lower concentration. Earlier, I told you, by the action of the sodium potassium ATPase, there is a high concentration of potassium within the cell when compared to that of outside, right? So there are n number of potassium inside this cell, and there are just few potassium outside. So eventually the potassium starts to move from a position of higher concentration to that of lower concentration. So this potassium starts to move out through this leaky potassium channels into the extracellular space. Okay, so eventually the potassium starts to move according to the concentration gradient. And now see, for one potassium lost, there is one negativity present. Okay, means inside the cell everything is electrically neutral. Okay, so for every potassium present inside the cell, there is an anion. Anion means negatively charged particle. So for one potassium, there will be one anion or one negatively charged particle. So the electrical neutrality is maintained. When these leaky potassium channels are open, or they are always present, whenever they are present, the potassium tends to move from a region of higher concentration to lower concentration. So the potassium starts to move out. Okay, leaving behind just the anion particles. Okay, so now what is happening? The potassium starting to move from a region of higher concentration to lower concentration, leaving back the anions. Okay, and earlier the sodium potassium ATPase was pumping out 3 sodium for every 2 potassium taken in. So both of them are contributing to this electron negativity, right? What eventually happens is that there will be a increase in the negative charge inside this cell. This sodium potassium ATPase, whenever it works, it causes the potential difference of around negative 5 millivolt. Okay, but whenever this potassium starts to move out through the leaky potassium channels to the extracellular space, this negativity starts to increase. It moves to negative 10 and then negative 15 and then negative 30. Eventually, it reaches around negative 85 millivolt. Okay, and when it reaches this negative 85 millivolt potential difference, or when it reaches this negative 85 millivolt potential, the inside of the cell becomes too negative. Okay, so that it is difficult for the potassium to move out, because here the negativity starts to attract the potassium. So potassium has a concentration gradient, and potassium here also has the electrical gradient. There are both of these forces acting. On one side, the electronegativity inside the cell, the negativity inside the cell starts to attract the potassium into the cell, whereas the concentration gradient is trying to take potassium outside the cell. But when the potential reaches around negative 85 millivolt, or when the negativity inside the cell reaches negative 85 millivolt, potassium is in a state where it cannot move into the cell or outside the cell. Okay, or in other words, at this potential, the concentration gradient is equal to the electrical gradient produced by the movement of potassium. Okay, or in other words, the concentration gradient for potassium is equal to the electrical gradient for potassium, and thus the net movement of potassium becomes zero. It cannot move into the cell or outside the cell. This particular potential where both the electrical and the concentration gradient are equal and there is no net movement of the cation is referred to as the Nernst potential. Nernst potential or the equilibrium potential. Okay. So here we can say the Nernst potential or the equilibrium potential of potassium is around negative 85 millivolt, because at this potential there is no net movement of potassium either into the cell or outside the cell, because both the concentration gradient and the electrical gradient are equal and they can cancel out each other. So now we will be discussing on our second hypothetical cell where the DNA encodes for a different protein. Okay. Here you can see we have the cell, we have the DNA, we have the sodium potassium ATPase doing its action. And what's its action? It pumps out 3 sodium for every 2 potassium taken in. Here consider the DNA encoding for a protein, and this time the protein encoded does favor sodium. The last time it favored potassium by providing the leaky potassium channels, but now it favors sodium by making the leaky sodium channels. And can you guess what is the action of leaky sodium channels? Exactly like the leaky potassium channels, they function the same. Okay, they allow the movement of sodium from a region of higher concentration to a region of lower concentration. So, as you all know that sodium is always in abundance outside the cell than it is present inside. Okay, so eventually sodium starts to move from outside the cell to inside the cell through these channels. Okay, and as discussed earlier in the case of potassium, this movement depends upon two factors. One is the concentration gradient of the particular ion. Okay, in the last case it was potassium, now it is sodium. Second case is the electric gradient. Okay, now the concentration gradient is favoring sodium. There is no much positivity inside the cell, thus sodium can easily pass through the ion channels into the cell. Okay, and this movement continues and eventually the cell starts to become more and more positive. Okay, so the potential of the cell starts to increase. It increases from 0 to 10 to then to 15, then to 30, and this continues till it reaches the 65 millivolt mark. Okay, when the potential inside the cell is 65 millivolt, the electric potential or the electric gradient inside the cell is too high. Okay, the concentration gradient is also high, but this time there is an electrical gradient which is also too high. Means inside the cell there is too much positivity and the cell is unable to accept sodium, because you know like charges repel each other. So when two positive charges come together, they start to move away from each other, they repel each other. So the same starts to happen here. When the amount of sodium or the concentration of sodium inside the cell increases to such a limit, it starts to repel away the external sodium. Means it doesn't allow the sodium to come inside. So this time the concentration gradient and the electrical gradient are both too powerful, and thus they do not allow the movement of sodium across the membrane. This potential, this 65 millivolt potential, or this potential where the net movement of sodium is zero due to the concentration gradient and electric gradient being equal to each other and cancelling out each other, is referred to as the Nernst potential or equilibrium potential for sodium. Earlier we discussed about the Nernst or the equilibrium potential for potassium. This is sodium. Okay, so what was the equilibrium potential or the Nernst potential for potassium? It was negative 85, whereas the Nernst potential for sodium is around 65 millivolt. So you might have heard about the equation called the Nernst equation. Okay, Nernst equation simply means an equation which is used to find the equilibrium potential of a particular ion. Okay, whether it be negatively charged, whether it be positively charged. Okay, this equation is used to find the equilibrium potential or the Nernst potential of the particular ion, which means the point or the potential where the net moment of that particular ion is zero due to the concentration gradient and electrical gradient being equal to each other and cancelling out each other. Okay, and this equation reads negative 2.3 RT by NF log to the base 10 concentration of the ion inside the cell by concentration of the ion outside the cell. Okay, R means constant, T means temperature, F means Faraday constant, N means the charge of the ion. Okay, so when we simplify all these constants, it becomes around negative 60 millivolt by N log to the base 10 concentration of the ion inside by concentration of the ion outside the cell. Okay, and the N is charge. So in case of potassium, N becomes 1. Potassium is plus 1, right? So potassium means plus 1. Concentration inside means around 4 or something, and outside means 140. And when we calculate this, we get approximately 85 millivolt. And similarly, it is a case of sodium too. You can use the values. These are some standard values. You can take it from some book or something and then calculate and see for yourself. Okay, this is what we call the Nernst equation. As the definition says, resting membrane potential is the membrane potential across the cell membrane when it is at the resting state. Means when it is staying simply, when we check the electrical potential across its membrane, it is around or it falls in the range negative 70 to negative 90. Okay, this is the range of membrane potential. See, earlier I told you the Nernst potential or the equilibrium potential for potassium was around negative 85, and the Nernst or equilibrium potential for sodium was around positive 65 millivolt. Okay, the resting membrane potential is around negative 90 to negative 70 millivolt. This is the range of the membrane potential, and this was practically proven. Okay, it's just a fact. Now see, so the resting membrane potential which falls in the range between negative 90 to negative 70 millivolt is much closer to the equilibrium potential for potassium, okay, than to the sodium. It is too far from the equilibrium potential of sodium. And what does it mean? It means most of the cells in our body are freely permeable to potassium than to sodium. Okay, so consider this cell. We have the sodium potassium ATPase working, and do you know, even though the resting membrane potential is negative 90 millivolt, the contribution of the sodium potassium ATPase towards this membrane potential, which is around negative 90 millivolt, is only negative 5 millivolt, around this range. Okay, so it doesn't contribute significantly towards the resting membrane potential of a cell, but it is the reason why there exists a resting membrane potential, because it causes the influx of potassium inside the cell and it pushes out sodium, and thus concentrating the inside of the cell with potassium and creating a concentration gradient for potassium to move from inside the cell to outside the cell. And thus this efflux of potassium from inside the cell to outside the cell is the reason why this negative 5 climbs up to negative 90 millivolt. Okay, so this is a main contributor for the resting membrane potential. And in some cells we have both the sodium and potassium leaky channels, and in that cases also the sodium contributes very much little, around negative 10 or negative 5, some range of potential to the resting membrane potential. So, and that's the reason why majority of the cells have a resting membrane potential of negative 90, which is closer to the Nernst potential or the equilibrium potential for potassium. And thus It just signifies one thing: the most of the cells in our body are freely permeable to potassium than to sodium, which is very less or negligible. Okay, this is about membrane potential and resting membrane potential. In the next video, we will be surely learning about action potentials and how is it different from resting membrane potential, and certain clinical scenarios that can affect the resting membrane potential and action potentials. Okay, till then, stay hungry, stay crunchy. And if you have any doubts or queries, you can surely comment it in the comment box below. Shows your support by subscribing to my channel and recommending this to your friends. Okay, till then, see you.